Hello, everyone. My name is Jonathon Hill, and I am a co-founder and vice president of science and technology at Wasatch BioLabs. And today I'm excited to talk about some work that we have been doing to use cell-free DNA, specifically looking at the epigenetic marks and methylation patterns within this DNA in order to diagnose neurodegenerative disease. So we are really working on bringing nanopore sequencing to the clinic, and we've developed a pathway in order to enable this process. The first thing that we need to do is sequence a whole bunch of DNA from different tissues of interest and build models that can predict the cell of origin of the cell-free DNA that is floating around in the blood. We then take that model based on our particular disease of interest, and we take an initial patient cohort and we test to see if that model is actually predictive of disease. Once we have a good proof of concept that this marker works, then we take it and we actually build on it to develop a commercially viable assay that can be used within the clinic. Then, of course, we go through the certification process and scaling in order to bring this to market. And so I want to talk about a project that is very much in process using this kind of pathway in order to identify biomarkers of Alzheimer's and Parkinson's disease and bring that into a clinically viable assay. And so what we did is we took purified primary cortical neurons, which are lost in Alzheimer's disease, as well as dopaminergic neurons, which are lost in Parkinson's disease. And then we sequenced them on a Promethean sequencer and compared them to blood plasma in general, which was pooled from multiple patients to give kind of a generalized background of what we would expect. After sequencing this DNA, we have developed two major tools to help us to analyze this data and identify regions that are differentially methylated between our different tissues of interest. The first of these tools is a file format that we're calling the CH3 format, and it is a very efficient data storage and transfer mechanism. And so what we take is what is typically 50 to 60 gigabytes of ModKit output, and we compress it down into something that's more in the 1 to 5 gigabyte range. This, of course, helps data storage on our server systems, as well as transfer to our customers. The second tool that we have made is called the Methyl Seeker package, and it is an R package with a number of functions for actually working with this data and analyzing it within the R framework. And so the pipeline that we used here is we took our DNA that we had sequenced from our different tissues. We used the CH3 format, but then what the package does is it creates a database on disk. And this is important because it allows larger than memory analyses to occur, which is very easy to run into issues with this amount of data that we're working with. We then conduct sliding window analysis to identify regions across the genome that are differentially methylated between cortical neurons in blood or dopaminergic neurons and blood. Because these tend to be regional effects, we then take those overlapping windows where we've identified significant differences and we collapse those into regions of differential methylation. So here's what we got with this particular case. We typically sequence to somewhere between 5 and 10x coverage. These happen to end up a little bit deeper than that, but you can see we do not need really deep sequencing in order to get results. Even at 10x coverage, we are essentially covering all of the CPGs within the genome. Because of this great coverage and because of the power and accuracy of the sequencing, we are able to identify 4.7 million differentially methylated CPGs between cortical neurons and the blood background, or 1.8 million CPGs between dopaminergic neurons and our blood background. Because these are regional effects again, we can collapse these into regions, and we have 224,000 regions between cortical neurons and blood plasma, or just under 150,000 regions between dopaminergic neurons and the blood plasma. Now we can take those regions as identifiable, measurable areas that can tell you if cell-free DNA came from a neuron or if it came from the blood, and we can go into patients. Now, with patients, we do not get a lot of cell-free DNA from a standard blood draw. We also want to make sure that we can have high throughput in our assay. And so because of these two factors, we typically sequence these at ultra low coverage. Within our study here, we had an average of 1/4 X coverage of the genome. So this creates sparse reads across the genome, and we can separate these into groups. There's first the cell-free DNA that falls into regions that are not differentially methylated between neurons in the blood, so they can't be used for a prediction. But a certain percentage of them, about 1 in 250 reads, falls within a region that we've identified as distinctive between our two tissues. And so we can use those for categorization as blood plasma background or neuronally derived cell-free DNA. And based on that, we can create an estimate of the fraction of cell-free DNA that is neuronally derived in our blood. So again, we use our methyl-seeker package in order to do this. We take our ultra-low pass cell-free DNA sequencing data, we go through the database creation, and then we summarize this time by read instead of by position. And this allows us to look at whole molecule analysis when we do these measurements. Then we can classify these reads based on our original data as neuronally derived or blood plasma derived and quantify those ratios of the cell quantifications. So we did this with a cohort of approximately 30 patients, some of which had been diagnosed with Alzheimer's, and then others that were in an aged matched control cohort and had not been previously diagnosed with Alzheimer's. You can see in the graph on the left that we have good separation in our data. There are 2 outliers in our control group that seem to have high fractions of neuronal cell-free DNA, even though they have not been diagnosed with Alzheimer's. We do not know at this point exactly why that is. However, one possibility is that these are actually pre-symptomatic or undiagnosed Alzheimer's patients. We know from epidemiological studies that in this age cohort, we would expect approximately 10% of patients to have some form of neurodegeneration, and this actually comes out pretty close to 10% of our control data set. We also had a few patients in here that had been diagnosed with mild cognitive impairment, but had not gone on to be diagnosed with Alzheimer's at the time of the study. So we compared our results in this analysis with 2 of the standard tests that are used to diagnose Alzheimer's in the clinic today. And you can see that if you look at our positive predictive value, even if we consider those 2 outliers as true negatives, that still produces an 89% positive predictive value. So that is a really good performance on par with the best tests that are out there. Our negative predictive value is 100% outperforming both of the other tests. And then we do have a small range where the distributions overlap. And so approximately 10% of our patients fall under an indeterminate range because they have kind of a medium level of neuronally derived cell-free DNA in their blood. So you can see overall our assay is performing as well or better than the existing assays that are on the market. We then took the exact same parameters in our model, applied it to our dopaminergic neurons versus blood, and then took a cohort of patients with Parkinson's disease and an age-matched control group again. And you can see in this one, the separation is also very good, even though we haven't customized the model for this particular use case. Positive predictive value and negative predictive value are both 100%, again, outperforming the tests that exist on the market today. Now, we have a lot of work to do going forward. We're, of course, trying to improve the performance and reproducibility by refining our model, trying to weed out regions that aren't that predictive, and determining the proper QC metrics, et cetera, for the patients that we sequence. We also are wanting to look at early diagnosis. The real goal of this test would be able to have a screening test where patients could find out if they are experiencing neurodegeneration long before they actually have symptoms, because this is when treatments are most effective. And so we have longitudinal data now that has tracked patients over a course of 20 years up until they were diagnosed with Alzheimer's. And we are sequencing that cell-free DNA that has been biobanked in order to get that pattern over time and see what that looks like before symptoms occur. We are also adding more neurons to this and trying to create a pan-neuronal panel in order to diagnose other types of neurodegenerative diseases. And of course, we want to make sure that this is high throughput and cost effective. And so we are also looking at targeting those informative regions and only sequencing the cell-free DNA that is going to be informative. This is just a little bit of a teaser because it's still in development, but we now are developing a method where we can sequence epigenetic marks in DNA in a targeted fashion. Most targeting methods use PCR, and so you lose any epigenetic marks, but we have developed a way to maintain those marks. And we can do it on genomic DNA as well as cell-free DNA. So this is going to be an exciting product offering coming out in the next few months. So with that, I just want to bring it back. As we've developed this assay for neurodegeneration, we really have built a platform that can be used in many different use cases for many different diseases. And we're excited to work with partners on applying these methods to other use cases to help bring nanopore technology to the clinic. So with that, I'll go ahead and quickly acknowledge our partners, the University of Kansas, as well as BYU, who helped in this research, and thank you for your time.